is kind of an underpowered study because we only had money to genotype 1,500 individuals, which for GWAS is very small. The last European GWAS that has been published had almost 40,000 participants with Parkinson's disease, and then about a million, more than a million healthy controls. We had 800 cases and 700 healthy controls, so it's very small compared to the normal GWAS. But we were, as I mentioned, we were able to see synuclein as a top candidate gene in our study, which is always good because that's the most consistent finding in both Europe and Asian GWAS. But we also found a little bit of signal on a gene, a variant close to a gene, the NROS gene in chromosome 3, which is a novel hit, and it comes mostly from Peru, from the individuals from Peru. As I mentioned, most people in Peru have about 75% of the genome that comes from Native American individuals. So it has a little bit of a European only. And then to be able to deal with this lack of statistical power to find things, we use kind of novel technique called Admixture mapping, where what we're trying to do is we use the ancestry from each of the variants, and we look at, instead of just a single variant, we look at haplotypes. So basically we split the genome, and if you do 23andMe or some of the other ancestry companies, you'll see that they give a report where you see each of your chromosomes, and parts of your chromosomes are colored in different ways depending on what the haplotypes or the variants are coming from. So they come from Europe or Africa or wherever. So that's called local ancestry. So you're looking at parts of your chromosomes that come from different ancestries. So we can do that with our individuals, and then we can, using bioinformatics, we cut the genome, and then we extract only the haplotypes that are non-European. So in Latin America, we see Native American, but we also see African ancestry, and we can take those two components and compare the frequency of those haplotypes in patients and healthy controls, and see if any of those haplotypes are overrepresented in one group versus the other. So that's a different way to do a comparison or an association study with disease risk. And using that, we identify several regions in the genome that are either associated to low risk for Parkinson's in Africa, and then high risk of Parkinson's in Native Americans. Those are novel results that obviously need to be validated in other cohorts. Unfortunately, there's no other cohorts in Latin America that can be used to replicate this. So we're just trying to build up our cohort so we can create our own replication sample. And then we also collaborated with 23andMe, and they did a replication of our top candidate genes, and they saw the same thing that we saw for synuclein. However, they were not able to replicate the new hit on chromosome 3. And we believe that it could be because their percentage of Native Americans on their Hispanics or their Latinos is much lower. Ours is on average almost 50%, and theirs is about 14%. I think their cohort is mostly Latinos that live in the US, so they're a little bit more ethnic with more European on them, and probably also they come from countries where they're more larger European ancestry. So